Dr. Nardi here, and in this video we are going to talk about how neurons communicate within each other. And if you remember back maybe to your biology course, you will remember that neurons communicate in electrical, chemical language. And what we're interested here is the synapse, or the connection between a neuron and another cell. The way it generally works is a presynaptic neuron transmits a signal, and the postsynaptic cell receives the signal. And the space between these cells is called the synaptic cleft or gap. And remember, no two neurons ever touch. In the 1920s, psychologists weren't sure whether neurons communicated at the synapse by electrical current or by releasing chemicals. However, a German psychologist believed that the synaptic transmission was chemical, but he did not know how to test his hypothesis. One night, he dissected out the beating hearts of two frogs and bathed them in a salt solution. He applied electrical stimulation to the vagus nerve attached to one of the hearts, which slowed the heartbeat. Then he extracted some of the salt solution, which he assumed would have captured any chemical that may have been released. When he applied this salt solution to the second heart, that heart slowed down too. This is just how he predicted it. Then he actually stimulated the accelerator nerve of the first heart, which caused the heart to beat faster. When he transferred the solution from the first heart to the second heart, the second heart sped up too. What he was on to that night, and he did not know it, was he was on to how neurons communicate by passing along electrically and by passing along information through chemicals. So we talk about them communicating in electrical chemical language. So the conclusion from his research were that hearts use chemical messengers, not action potentials, to change the heart rate. How this works is neurotransmitters are stored in terminals in the membrane enclosed in bubbles called vesicles. Think of them as little bladders. That's actually what the term means. And when an action potential arrives at the terminal, it opens the channel that allows calcium ions to enter the terminals from the extracellular fluid. The calcium ions cause the vesicles clustered nearest to the membrane to fuse with the membrane. The membrane then opens, and there neurotransmitters spill out and diffuse into the cleft, a process we called exocytosis. So the action potential causes exocytosis. Ionic tropic receptors form an ion channel that open quickly to produce an immediate reaction required for muscle activity and sensory processing. Metabotropic receptors open channels indirectly through a secondary messenger, and they act slowly and produce longer lasting effects. And opening these channels is what sets off gradiated potentials that initiate an action potential. On the postsynaptic neuron, the neurotransmitters dock with specialized protein receptors that match the molecule shape of the transmitter molecule. Think of neurotransmitters like a key and the receptor sites like a lock, and only certain receptor sites can open certain locks. And we have ionic receptors. These receptors cause ion channels to open, which has a direct and rapid effect on a neuron. We also have metabotropic receptors. They open channels indirectly, producing slower but longer acting effects. Please note that synaptic transmission is much slower than the axonal or electrical transmission. Activation of receptors on the postsynaptic cell have two possible effects on the membrane potential. Hypopolarization. This creates an excitatory postsynaptic potential. We call these EPSPs, and these open sodium channels. This process makes the postsynaptic neuron more likely to fire. So we refer to these as, again, excitatory postsynaptic potentials. Hyperpolarization creates an inhibitory postsynaptic potential, IPSP, and the IPSP opens potassium or chloride channels, or sometimes both, and this makes it less likely an action potential will occur. There's a couple things to think about when we think about EPSPs and IPSPs. They are graded potentials. They accumulate over a short time, so there's some kind of temporal summation going on where they receive information at different time points, and they also combine inputs from different locations on a dendrite. We call this spatial summation. And so all this information is being gathered, and think about the neuron itself acting like an information integrator or doing some kind of summation. And the neuron is basically the decision maker, and it gets the excitatory information, it gets the inhibitory inputs, and it does some kind of algebra, and when that threshold is met, it will fire. Neurotransmitters must be removed from the synaptic cleft in the space around the synapses. This will allow for more frequent responding, and it will prevent these neurotransmitters from affecting nearby synapses. There's different processes involved, but one is reuptake. The transmitter is brought back into the terminals, and there's also inactivation. Enzymes will break down the transmitters in the cleft. Interestingly enough, if we talk about drugs, some drugs mimic natural transmitters and stimulate receptors. We call them agonists. Some block neurotransmitter receptors. We call them antagonists. And some enhance or reduce transmitters' effects. For example, antidepressants block the reuptake of serotonin. We call these selective serotonin reuptake inhibitors, or SSRIs. Some prevent the neurotransmission inactivation, and these are MAOIs. Glial cells, they prevent transmitters from spreading to other synapses. They also absorb and recycle transmitters from neurons' reuse, and they also release glutamate to regulate the presynaptic transmitter release. Now, we could talk about receptor subtypes, and this actually gets to a more complex conversation. For example, acetylcholine receptor has a nicotine and a muscarinic subtype. Also note that neurons can release more than one chemical. One fast-acting plus one or more slower-acting neuropeptides. They can release two or more fast-acting transmitters. They can release an excitatory and inhibitory neurotransmitters at different synapses. And again, it gets very complicated very quickly. Just know that this whole process is very, very complex, and we're only scratching the surface. Basically, neural networks add further complexity to the neural process. Think of these as groups of neurons that function together to carry out a process. If you want to look more into this, I would look into the Human Connectome Project. This is a large-scale, multi-university effort to map the brain circuits. It's fascinating. Anyway, hope you enjoyed this video, and I'll see you next time. Take care.